Hello everyone, I am Adham Amir, and today inshallah I will be revising with you the Lipid Metabolism chapter, me and my colleague Shazia. So I will be covering the first part of the chapter, which is the triacylglycerol part, plus the fatty acid degradation in general. So before I start, I would like to point out some notes or stuff I would like you to know. So first you will realize that I'm using a lot of pictures from the book, and this is to tell you that these pictures are very important. They can ask about the smallest details, even the one written with a small font below these pictures in the book. Also, they are very good visuals that you can use to memorize and understand the um, reactions, not only in this chapter but in the whole biochemistry course. So let's start. So I would like you to know that acetyl-CoA is a key player in lipid metabolism, and you are going to see how much we are connecting different pathways and reactions and molecules using acetyl-CoA. So it's very important key player in lipid metabolism, as it is a substrate for fatty acids and steroids formation, as well as the product of their degradation. So you know, of course, that fats in general is the most important storage form for energy in our body. So acetyl-CoA connects this storage form, which is fats in general or lipid, to the different types of molecules in order to make them produce energy. So as you can see, they are important, and they connect the lipid to the citric acid cycle, which is going to be the way we produce energy from lipids, as we are going to see. So let's start with absorption. Before we do anything with the lipids, we need to absorb them, because most of the fats in our body is absorbed through food. As you can see, triacylglycerol, one second. Okay, triacylglycerol or fat is a big part of our diet. It is absorbed in the lumen of the small intestine, which is called the exogenous pathway. So the exogenous pathway is the fats that are acquired from the diet or the food. Why it's called like this? Because it's from outside of the body. There is also the endogenous pathway, which is when lipids is produced by the liver, but we rely on the exogenous pathway more, and it's more important. Of course, both are important, but exogenous pathway um, is used more by the body. So let's start from the beginning. Dietary fat first passes through the lumen of the intestine, as you can see here, intestine, and then it's going to be mixed with bile salts. Sorry, before I continue, I would like you to know that biochemistry may seem hard in the beginning. I also thought like this, especially when I started the Biochem 2 course. But actually, it will become much easier if you understood before you try to memorize. So don't try to memorize at all before you understand the concept and understand the flow of the pathway. What do we need from this pathway? Like before memorizing anything, just ask yourself, what do we need from this dietary fat? So how can we achieve it? And with time, you're going to see that the enzymes names, which is also very important to memorize, is going to be even like predictable for you. Even if you don't memorize it 100%, you can predict it if you want, if you know the type of reaction and what's happening in the reaction. So let's continue. So first of all, dietary fat will be mixed with bile salts, okay, and these bile salts will cause the triacylglycerol to produce emulsion of triacylglycerol. This emulsion is going to be broken down using pancreatic lipase, and from the name pancreatic, it's produced by the pancreas, and lipase, which means it will break down this lipid, which is triacylglycerol, into 2 fatty acids and 1 monoacylglycerol. Okay, structures are not important to memorize in this. Most of the structures are not important to memorize, but I will also I will always refer to the structure in order to understand. So whenever I'm talking about any structure, it's not for you to memorize, but it will help you understand the process more. So of course, from the name triacylglycerol, it's formed from one glycerol molecule and 3 fatty acids. So here it's broken down to monoacylglycerol and 2 fatty acids. Why is this step important? Because although the membrane, the cell membrane of the enterocytes, which is intestinal cells, are also hydrophobic, but due to the huge size of triacylglycerol, they cannot pass this membrane. That's why we need to break it down to smaller parts, 2 fatty acids and monoacylglycerol, and then they can pass through the membrane. Whenever they pass through the membrane, they will mix again or join again together to form triacylglycerol. Also, another important like factor here, which is going to be also repeated in this chapter a lot, is the activation of the fatty acid. As you can see here, fatty acid is first joined with CoA, which is the coenzyme A, to fatty acyl CoA, and then it's joined again to monoacylglycerol. So what is important from this is the concept itself. Fatty acids alone are inactive, are inert. They are not seen by the body. They won't participate in any type of reaction. So we always need to add CoA to the fatty acid to produce fatty acyl CoA. This process we are going to also highlight it again. It needs ATP, so let's not worry about it now. But that's why it's joined with CoA before it forms triacylglycerol again. So these triacylglycerol are now in the enterocyte cell, needs to be transported to the adipose tissue to be stored, for example. So how are they going to be transported? So they will form huge balls called chylomicrons with cholesterol, phospholipids, triacylglycerol, of course, and ApoB48, which is produced by liver. This huge ball of chylomicrons will travel through the lymph nodes and then through the blood to adipocytes or the target tissue in the body. Okay, so these chylomicrons, in order to be absorbed, they need some enzymes and some key players to help it to be absorbed properly. First is LPL, or lipoprotein lipase enzyme. It separates the glycerol from the fatty acid. So as we said, it's triacylglycerol, which is forming most of the chylomicron. So whenever it reaches the adipocytes, only the fatty acids will enter the adipocytes. The glycerol will stay in the chylomicron. So this LPL will separate them in order for the fatty acid to go inside the adipocytes and the glycerol to stay in the chylomicron. We have something called ApoC2, which activates LPL, which we just talked about, and ApoE. Okay, after the fatty acid enters the adipocytes, now we have glycerol in the chylomicrons. These chylomicrons are called chylomicron remnants, and they have to be cleared because they are in the blood right now, and they are of no use now after we transported the fatty acids into the adipocytes or whatever the target tissue was, but mainly adipocytes for storage. So the ApoE will stay on the chylomicron as well, on the chylomicron remnants now, and the liver will detect the ApoE and will understand that this is a chylomicron remnant. So now it needs to be absorbed by the liver. So this ApoE is just for marking this remnant to be cleared by the liver after it's done. So the chylomicron now is we can say it's like the taxi for the fatty acids to the adipocytes. Of course, remember that chylomicrons are only needed for long chain fatty acid, which are longer than 12 carbons. But any fatty acid, as you can see here, the triacylglycerol structure I just talked about, any fatty acid less than 12 carbons, which is called short chain fatty acid or medium chain fatty acid, we are going to use, or not we, but the body is going to use the albumin, which is a blood protein, to transport them. So short chain fatty acid is transported directly through albumin through blood. Okay, here is also a summary for the triacylglycerol metabolism. It produces fatty acid, as we saw, which is the most concentrated energy source in the body. Okay, so it will either be degraded to produce energy or used in membrane synthesis. Of course, you know that membranes are mostly lipids. They are phospholipid bilayer, for example, in the human cells. We are going to see this as well. So, fatty acid or the fatty acids are going to be degraded when we need energy. And what determines if the fatty acid will be stored or degraded? It is the cell need. It is the body need. It is a cell metabolic needs. Okay, so triacylglycerol metabolism, this part is logical. It compares when will the triacylglycerol be stored and when will it be degraded for energy. Right after a meal, insulin is high, of course, as you know, due to raised blood sugar, which will inhibit hormone sensitive lipase, which is an enzyme we're going to see in the next slide, which degrades not triacylglycerol, which degrades fats in a certain type of fat we're going to see now, and promote the synthesis of LPL. If you remember, LPL is used to absorb the chylomicron by separating the glycerol from the fatty acid, plus activating the production of triacylglycerol. Okay, so it activates the production of triacylglycerol for storage. Okay, but when blood glucose level drops after some period of fasting or without any source of energy entering the body, glucagon level increases, which is opposite of insulin, which causes net triacylglycerol hydrolysis, which means we are breaking down triacylglycerol for energy production. Here it is showing the exogenous and endogenous pathways and the cycle of lipids in general in the body. So as you can see, some are produced by the liver, some are from food. Okay, so here is the important reaction I told you about, which is activation of the fatty acid. As you can see here, it can be any fatty acid. It's activated by adding CoA to it. But in order to add CoA to the fatty acid, we need ATP. We need an energy molecule here. Although it is only shown as one ATP, it is considered to be 2 ATP equivalent, because as you can see in the products, it is then transformed into AMP, not ADP. So we say that activation of one fatty acid uses 2 ATP equivalent. So you are going to ask me, why do we need to know how to activate fatty acids? Now, because we knew how to break down the triacylglycerol for transportation. We also knew how to let them pass through the enterocytes. They are now sitting happily inside the so the fatty acid, as we said, only the fatty acid passes the chylomicron into the adipocyte. So we need to store it as triacylglycerol, not as fatty acid alone. So now we need to know how to form triacylglycerol from fatty acid. So the first step is activating it, as we just said. Now, after we activated it, we need a glycerol, right? Because it's triacylglycerol. We can get it with multiple ways. First of all, here you are going to connect with glycolysis or carbohydrate metabolism in general. You should remember, if you remember. So here we have glucose. It will be broken down until you reach dihydroxyacetone phosphate, which is glycolysis. From here, we will take this dihydroxyacetone phosphate. We don't have to continue until glycolysis ends. We can take it from here and start using it to produce glycerol, for example, by a dehydrogenase. So, of course, you should know by now that any dehydrogenase enzyme is for oxidation reduction reactions. So, as you can see here, it gained hydrogen, which is reduction. So here we have a reduction process to produce glycerol 3-phosphate from dihydroxyacetone phosphate. Okay, we can get this glycerol 3-phosphate from different ways. For example, if we have glycerol by adding a phosphate by a kinase, it will directly transform into glycerol 3-phosphate. We can also use a process called glyceroneogenesis, which is similar to gluconeogenesis. It's actually the same thing, but the only difference is that instead of forming glucose from non-carbohydrate precursors or molecules, we are forming glycerol 3-phosphate from non-carbohydrate molecules such as lactate, pyruvate, and some amino acids. Okay, so after we got the glycerol 3-phosphate by a reduction reaction, now we are going to add a fatty acid to it, because I need as I told you, you need to understand what is the purpose from all of this. The purpose, as we said now, is to form triacylglycerol for storage. Okay, so we need this glycerol 3-phosphate to be this glycerol to have 3 fatty acids connected to it. So we are going to simply add 1 fatty acid by glycerol 3-phosphate acyltransferase. Acyl means fatty acid, and transferase is just transferring it. So it's transferring a fatty acid into this glycerol 3-phosphate, producing glyphosphatidic acid. What is glyphosphatidic acid? It is a glycerol with 1 fatty acid and 1 phosphate connected to it. So before we proceed, we are going to see the other side, which is also available in the peroxisomes, for example. The dihydroxyacetone phosphate will be added to a fatty acid before we reduce it. So the acyltransferase will be first. So the enzyme name will be dihydroxyacetone phosphate acyltransferase. Now we added the fatty acid to it. So we need to reduce it in an acyl dihydroxyacetone phosphate reductase. Here it's also a reduction reaction, but it's using NADPH instead of NADH. Of course, you will need to memorize these as well. So we will end up with lysophosphatidic acid. Lysophosphatidic acid, as I said before, simply a glycerol with a phosphate group and a fatty acid. So what is missing in order to transform it into triacylglycerol? 2 fatty acid, right? So we are going to add 1 more with acyltransferase again, producing phosphatidic acid. Here we can stop and take this to produce phospholipids, which is important for membrane synthesis and other stuff if the body needs, or we can continue. Of course, we will continue because we need triacylglycerol. So we will continue, but there is a problem. Now the 3 carbons are occupied, 2 with fatty acids and 1 with phosphate, and we need this phosphate to go in order to free up space for the 3rd fatty acid. So we will use phosphatidic acid phosphatase. The enzyme names are very easy because it's phosphatidic acid and phosphatase. What is phosphatase? It's just removing of the phosphate. It will give us diacylglycerol because now it's only glycerol with 2 fatty acid, and then we will add the 3rd one was also an acyltransferase. So this is a complete picture of forming triacylglycerol in order to store them. Okay, so now here is the glyceroneogenesis that I told you about. We can get it from different precursors like lactate, alanine, which is an amino acid, pyruvate as well. So here it is just fast revision. Of course, this picture is not from the book, so sometimes some enzymes are in different names. So I just put it to remind you that we got the dihydroxyacetone phosphate. We can get it from glucose, and then we use it for triacylglycerol formation. Okay, now the body needs energy. We are needing energy, so we need to use this triacylglycerol we just stored. So the story will start with what? The story will start with glucagon because it is the indicator that the body is needing energy. Okay, so the hormone receptor interaction, the glucagon will interact with this receptor, which will start a whole cascade in order to, which will like send a signal to start triacylglycerol hydrolysis. So first, it will activate the cyclic AMP, or with adenylate catalase, it will use 1 ATP to produce cyclic AMP. This cyclic AMP will activate PKA, which is protein kinase A, this pKa will activate perilipin 1. Okay, so it will activate perilipin 1 by phosphorylating it, as you can see here. And this perilipin 1 will activate CGI58, which will then start degrading the triacylglycerol. How are we going to degrade it? Using 3 enzymes, because we have 3 fatty acids. The first one is ATGL, which is adipose triacylglycerol lipase. Adipose because it's stored in adipose tissue, triacylglycerol because we are breaking down this molecule, and lipase, as we said, is breaking down of lipids in general. This will result in diacylglycerol and 1 fatty acid. And then we have HSL hormone, or enzyme, which is hormone sensitive lipase, is going to break it down to monoacylglycerol and fatty acid. And then MGL, monoacylglycerol lipase, will finally give us the 3rd fatty acid, and then glycerol. So now we broke down triacylglycerol successfully. How about insulin? Now the body doesn't need energy anymore, so of course insulin is being produced. Let's say you ate, so you don't need to keep breaking down triacylglycerol. So insulin, of course, we know it's the opposite of glucagon. So what is it going to do to stop breaking triacylglycerol? We have 2 ways to stop the hydrolysis. First one is by binding also or activating PKB, which is protein kinase B. It will inactivate the cyclic AMP. So it will stop this pathway, we can say, from the beginning. But if we just stop it from here, we still have perilipin 1, which is active. We still have perilipin 1, which is already active. It will keep breaking down. So the second part, So the second part which is going to be inhibited is the perilipin 1 itself. As you can see here, the insulin will activate a phosphatase enzyme, which will remove the phosphate from the perilipin 1, inactivating it. So now we stopped completely, or not completely, but we stopped to a very big extent the hydrolysis of triacylglycerol due to insulin. Okay, so now we are done with triacylglycerol degradation, transportation, and formation, which is mainly explaining how do we store triacylglycerol and how do we move it through the body. Now, we still didn't produce any energy. Of course, we still didn't produce any energy or anything. And the main importance of lipids, as we said in the beginning, it is the most concentrated form of energy storage in the body. And of course, we all know that lipids is the most important energy source reserve. So how are we going to produce this energy? From the fatty acids. So now, in the previous step, we destroyed or we hydrolyzed the triacylglycerol into 3 fatty acids and 1 glycerol. These 3 fatty acids we are going to use to produce energy, which is called beta oxidation. We're going to know now why it's called like this. So before we start, I would like also to introduce the structure of fatty acids in general. As I told you, we don't need to memorize any fatty acid structure, but it's just for understanding, and it's easy. It doesn't need memorization. So we have carboxylic acid group, which is C double bond O with OH, and we have a long hydrocarbon chain, which is just carbons with hydrogens. And these number, or the number of carbon, when it differs, for example, we have 16 carbon fatty acid, 18 carbon fatty acid. Of course, it gives us different fatty acids. So before we start, I would like to highlight the naming of the carbons. So the first one was the carboxylic group. It doesn't have a name, and starting after it, we will start counting alpha, beta, gamma, and etc. So this one is the alpha carbon. Although it is not the first one, because the first one is here with the carboxylic group, but this one is called the alpha carbon, alpha, beta, and so on. And the last one is always called omega. There is also something called omega oxidation, but we are not going to study it because It's more important and more used in animals, not in humans. But the last one is always called omega, despite it being the 16th carbon, if it's a 16 carbon fatty acid or 18 carbon fatty acid or whatever. So now we want to focus on the beta carbon and the beginning of the fatty acid. So okay, 1 second. So before we go into this, as I said in the beginning, acetyl-CoA is the most important key player in lipid metabolism in general. So let's introduce also the acetyl-CoA structure. So do you know what is acetyl-CoA? It is 2 carbon, of course we cannot see it here, but it's 2 carbon with a CoA. So only 2 carbon with CoA, which is coenzyme A, is called acetyl-CoA. Okay? So our purpose here is to produce acetyl-CoAs. So as I just said, for the structure of the acetyl-CoA is just fatty acid, which is all of this, for example, plus CoA, which is the coenzyme A. Here also is the structure. So beta oxidation, from its name, it cuts before the beta carbon. So if we said we are going to beta oxidize this structure, of course this structure cannot be beta oxidized because it's not activated. This is a structure for fatty acid alone, but just I want to illustrate on it. We said this is the alpha carbon, this is beta carbon. We will break here. We will break here exactly, which is between the alpha and the beta carbon. Okay? We will break here, and this part will go, of course it will be connected to CoA as well. 1 second. It will be connected to CoA as well, because we said it have to be activated. CoA. So yes, here, so the final result is going to be 2 carbons with CoA. And what do we call this molecule? We call it acetyl-CoA. Acetyl-CoA. It's written here, acetyl-CoA. It's very similar to acyl-CoA when it comes to how it sounds, but they are totally different stuff. Acyl-CoA is a general term for fatty acid, activated fatty acid, which is fatty acid plus CoA. So it can be any number. It can be 16 carbon fatty acid with CoA, 18 carbon fatty acid with CoA, but acetyl-CoA is a specific structure, which is 2 carbons with a CoA. Here is the correct structure, but I tried to illustrate it here as well. 2 carbons, as you can see, with CoA. This is called acetyl-CoA. So this is beta oxidation simply when it comes to the idea. But if we if it was that simple, we would lose all the other carbons. Why? Because as you can see, first of all, we took the CoA, and the rest of the structure doesn't have CoA anymore, so it's inactive. So we took 2 carbon, which is acetyl-CoA, and we will know in the following slides how we are going to produce energy from it. But we will waste all of the other carbons because they are not activated. And also there is, as you can see here, there is a carboxylic acid group. So we need this bonding to happen. Like we need a COO in the rest of the structure. Like here, in the new formed structure after we separated it, we need it to have C double bond O. So before doing this step, we will have to prepare it first. Beta oxidation is 4 steps. The first 3 are just for preparation. The fourth one is the one I just told you. We'll just break before the beta carbon, and we will form acetyl-CoA, which is a very important molecule we needed to produce energy. So let's start. Acetyl-CoA is any, as I said, fatty acid with CoA. I know I repeated this a lot, but because I had hard time discovering this, that's why I want to make sure I are not confused. So acetyl-CoA. Okay, so acetyl-CoA, we start with it. Of course, you need to memorize all of these. The FAD, H2O, and like this. So I memorized it by Actually, this was told to us as well when we were foundation, and I remember it till now. In order to memorize the sequence, like is the FAD first or NAD plus, like this, I memorized it as FAD my NAD os. FAD FAD my, which is water in Arabic, NAD NAD, and then os yani cut. So because here we will separate the two molecules, it will cut. So this is how I memorize it, like in a short form, in order to recall it fast, especially the arrangement of the reactions. So the first one will use FAD, which is FAD, by acyl-CoA dehydrogenase. As I told you before, dehydrogenase, oxidation or reduction reactions. Here it is losing or yes, getting rid of hydrogen, which is oxidation. So the first oxidation reaction here, it will form trans alpha beta enol CoA. What did we do here? We simply formed another double bond. Why? Because we will be taking one with the acetyl CoA, so we need another one to stay with the rest of the structure. Okay, so enol CoA, we will take it. We will take, for the enzyme naming, it's easy. We will take the enol CoA hydrase. So it is hydrating the enol CoA molecule by adding H2O. Here it will form beta hydroxyacyl CoA in the L form. Okay. Now we are going to oxidize it again, but this time with NAD. One second, let me. This time with NAD, we are going to oxidize it again, but the enzyme name will change because the substrate changed. Here it was acyl CoA, so we just said acyl CoA dehydrogenase. Here it's beta or L beta hydroxyacyl CoA, so we will say L beta hydroxyacyl CoA dehydrogenase. As simple as that. Another oxidation reaction, and now this oxidation reaction will form a double bond between the beta carbon. This is a beta carbon. This is a carbon with the CoA. This is the alpha carbon. This is beta carbon. So we form double bond in the beta carbon, and now it's ready. And the name of this molecule is beta ketoacyl CoA. Now it's ready. Why is it ready? Because if we cut here right now, if we cut here like this, this is خلاص, the blue part here is acetyl CoA. If you cut here, the rest of the structure have the C double bond O. So thiolase, which is very important enzyme in this reaction, will do two things. First of all, it will cut here, so it will give us acetyl CoA. But now we have another problem, which is that the rest of the structure, yes, now it has a double bond, a C double bond O, but it doesn't have a CoA. So if it's left like that, it will also be wasted because it's not activated. So it will add a CoA to the acyl to produce acyl CoA. So now we have acyl CoA and acetyl CoA, two CoAs. One, it was already here from the beginning, and the other one was just added by the thiolase enzyme. So thiolase enzyme has two functions. One, it cuts to produce acyl CoA and acetyl CoA. Two, it adds CoA to the acyl to become acyl CoA. So now we are going to say we started with acyl CoA and we ended with acyl CoA plus acetyl CoA. So how come we just like produced something from thin air? It seems like this, but no. This acyl CoA, for example, if it was 16 carbon in total, this one it became 14. Why? Because we took 2 carbon to the acetyl CoA. So this one is actually not the same as this one. That's why it's not called beta oxidation cycle. Because cycle, what we start with, we have to end with exactly. No, but it's called instead a beta oxidation spiral or beta oxidation rounds. Because what we start with, we end with something different, but it's very similar to it. That's why there is a difference in naming. Here also we have what I just said about all of the reactions, but also in addition to more important details. Of course, you have to. This is a beautiful thing about biochemistry, that you have to connect all the reactions together and all the chapters together. So here, if you remember from the TCA cycle chapter or chapter 3 in biochemistry 2, as we said, we just produced FADH2. Okay, so don't be tricked. This FADH2, how is it going to produce energy? You already know how to produce energy from FADH2, but don't be tricked. It won't be used by complex 2 because this is a trick part. It can be asked as a trick question. So be aware. This FADH2 won't be used by complex 2. Sorry, it's not in TCA cycle. I meant in electron transport chain, in ETC, not TCA. Sorry. So it won't be used in complex 2 because the FADH2 which is used in complex 2 is the one coming from succinyl dehydrogenase. Okay, and does this come from succinyl dehydrogenase? No. The enzyme is acyl-CoA dehydrogenase. It's a different enzyme. So it doesn't match, so it's not going to be used by complex 2. So what is going to happen? Are we going to waste this FADH2? No. We are going, it is going to be carried by ETF, which is electron transport flavoprotein, to ubiquinol, UQ. So this one is not going to be used by complex 2, it's going to be used by ubiquinol and transported by ETF. So don't be tricked by this detail, it's very important. Here we just said it is a hydration reaction, breaking down of the double bond. The enzyme is called NOL-CoA hydrase. Here also we need to know what is going to happen, what is the phase of this NADH. This NADH is going to be used normally to produce energy. It's going to be donated to complex 1. So make sure that you connect all of the chapters together and all of the reactions together, because in real life the cell doesn't know that lipids is put in a different chapter than ETC, than carbohydrates. It's all working together, so you have to treat them as one unit. Here as well, we can see that the thiolase enzyme is also called beta ketoacyl CoA thiolase, which is just thiolase, but before it is a substrate name, which is ketoacyl CoA. Also, this step is called thiolytic cleavage, and from the name, thiolytic and cleavage is just the cleavage of the molecule. Okay, so now we are done finally cutting the fatty acid into pieces. But as I said, we have different number, we have different number of carbons in different fatty acids. We have 16 carbon fatty acid, 18 carbon fatty acid, I don't know, 21 or 20 carbon fatty acid. So of course, different fatty acids produce different number of molecules and different amount of energy. So here we are going to take an example to know how to calculate how many beta oxidation rounds are needed and how many energy is produced, or how much energy is produced. So the example is going to be on palmitoyl-CoA. Palmitoyl-CoA is actually, why is this one used as an example? It is the most common fatty acid found in nature, and it is a 16 carbon fatty acid. So if I told you how many beta oxidation round we need to completely break it down, don't be tricked into dividing it by 2. Like you say 16 divided 2 because we remove 2 carbons each time is 8. No, let's do it very fast. 16 minus 2 will give us 14. Minus 2, 12. Minus 2, 10. Minus 2, and if you do it enough times until you reach the last one, when it's 6 minus 2, 4. 4 minus 2 equal 2. So the last one won't be like broken down again. So we actually need only 7 rounds, because the last 4 carbons will be broken into 2 acetyl-CoA, and we won't need an 8th round to break down anything. Okay? So it will always be 1 less round than the half. For example, 16 divided 2 is 8 minus 1. If we have 18, if we have 18 carbon, 18 divided 2, 9 minus 1. So it will only take 8 rounds to fully destroy, or not destroy, hydrolyze or oxidize. So it takes 7 rounds for a 16 carbon fatty acid. And what will it produce? It will produce 7 FADH2, because with each round it will produce 1 FADH2, and with each round it will produce 1 NADH, so 7 and 7. But as we said, it will produce 8 acetyl CoA. So the only thing you can find by dividing by 2 is the number of acetyl CoA. Okay, as you can see here, it's also the summary of what I said. You will need 7 water molecules, don't forget, because we use 1 in each round. We need 7 CoA because we use it in the last reaction, if you remember. Okay, so now let's calculate how many energy, because till now I'm telling you we are producing NADH, FADH2, acetyl CoA, but the body needs energy. It doesn't need acetyl CoA or NADH or FADH. They are just energy molecules or energy carriers. So we need to translate them or like produce energy from them. So remember, 1 acetyl CoA gives 10 ATP in TCA cycle, if you remember. How? Because 1 acetyl CoA will produce 3 NADH and 1 FADH2 through TCA and 1 direct ATP. If we converted NADH to ATP, of course, you should know 1 NADH is equal to 2.5 ATP and 1 FADH2 is equal to 1.5 ATP. So when we multiply the numbers, we will get 7.5 from NADH, 1.5 from FADH, and 1 like direct ATP. So we will get the 10 ATP from 1 acetyl CoA. So let's return to the palmitoyl CoA. We said we got 8 acetyl CoA. Multiply by 10, we have 80 ATP. But don't forget the FADH2 and NADH as well. We have 7 NADH, multiply 2.5, we will get 17.5 ATP. 7 multiply 1.5, which is the FADH2, we'll get 10.5 ATP. If we added all of these, we will get 108 total ATP from a 16 carbon fatty acid. But this number is still not the final or the actual ATP we got, because if you remember the first step in the it was in the carnitine shuttle slide. The first ever step when we took the fatty acid and we started working with it, we added CoA, if you remember, with the first step in the carnitine shuttle as well, which is the carnitine acetyltransferase 1, CAT1. It added CoA to fatty acid, activating it using 2 ATP equivalent. So we will subtract 2 ATP equivalent to get the net ATP. So 106 net ATP from the 16 carbon fatty acid. If we did the same calculation with 18 carbon fatty acid, we will get a different number. Of course, we will get more because we have more carbons, but it will be different. And so on with different numbers of carbon. Now we want to compare it to carbohydrate metabolism. Why did we say that lipids is the most efficient or the best way to store energy? Because if you compare it, if you divide the number of ATP we get with the number of carbons here, and in glucose, for example, let's say we get 31 by 6 carbons, we are going to get 6.6 ATP per carbon, but here only 5.5. So you can see that lipids is much more effective in storing energy, because when we break them down, they give us more energy, indicating they are better storage and they are better, like they are holding energy better. Okay, here also I just put this in order to remind you with something that helped me in Biochem 2. You have to write, of course, if you are suffering with memorizing, you have to write the reactions with your own hand. As you can see, my handwriting isn't the best, but for me, this helped me more than the book. Of course, I copied it from the book. I didn't invent anything new. Yeah, if you remember the picture I just showed you, it's the exact same thing. But whenever I write it with my own hand, like, it is easier to remember it in the exams, and it's easier to memorize it in your own handwriting. So I really recommend you writing everything, at least not everything, at least the reactions, with every details like the enzyme name, the FAD, everything. It will help you a lot. Okay, so now we are finally done with fatty acids, the normal fatty acids. But we have different types of fatty acids. We have four special cases we are going to deal with now, which is the last thing in degradation, which is a big chunk of this chapter. So the one we were just degrading and getting energy from were saturated fatty acid. Saturated means they only have single bonds. Of course, not the C double bond O. I mean single bond in the long chain hydrocarbon. You remember the CH. It was all single bond, which means it was all single bond, which means saturated. Okay, so let's continue. Okay, so we have special types or different types of fatty acids which may change a bit in the degradation process in order to produce. Okay, so the first type of special fatty acid, they are not special but different than what we just dealt with, is very long fatty acid chains. For example, if we have 24 carbons fatty acid, 26 carbons fatty acid, how are we going to deal with them? Actually, we are going to deal with them in a very similar way, but with a few differences that we have to know. So these type of fatty acid are called very long fatty acids chains. Okay, so they will be, or the degradation for them will start in the peroxisomes. That's why it's called simply beta oxidation in peroxisomes. So it is the same process but in a different place, because the mitochondrial enzymes cannot work on very long chain fatty acids. So we have a different way in peroxisomes, where peroxisomes can work on only very long chain fatty acids. So let's start. First of all, it will remain the same beta oxidation that we know, but we will have few differences. First of all, it uses peroxisomal carnitine acetyltransferase. Do you remember the CAT enzymes we just talked about? We said CAT1 and CAT2, which is abbreviation for carnitine acetyltransferase. We will just add the word peroxisomal before it. So instead of it being called carnitine acetyltransferase 1, for example, we will call it peroxisomal carnitine acetyltransferase 1, which is logical to differentiate it between the one in the mitochondria. This is one of the differences. And of course, this one only works on the very long fatty acids. So peroxisomes only work on the very long fatty acids, and the mitochondrial works on the rest. So the same reactions are as normal, with some exceptions. First of all, do you remember the second and third steps? Yes, the one with the annual CoA hydrase and the beta hydroxyacyl CoA dehydrogenase. The second and the third one, the hydrase and the dehydrogenase, but the second one was the NAD plus. They are found on a single molecule. So instead of going to the hydrase and then it forms step 2, going to the dehydrogenase and forms step 3, they are both found in one big molecule. So it does step 2 and 3 like right after each other, you know, like it's like a shortcut. Both are found in the same place, instead of going to two different places, let's say, to do two different jobs. So you have to memorize and know that the second and third step are found in the same molecule. The second and third step enzymes. So the hydrase from the second step and dehydrogenase from the third step are on the same molecule. Okay, the first reaction is catalyzed by a different enzyme. If you remember the normal beta oxidation, the first enzyme was acyl-CoA dehydrogenase, which used FAD and then produced FADH. But here it is acyl-CoA oxidase. It's a different enzyme, and we are going to see how will this affect now. But you have to know. So the first enzyme is totally different. It's called acyl-CoA oxidase instead of acyl-CoA dehydrogenase. The second and third enzyme, they are the same, but they are on the same molecule as well. So you can memorize it like this: the second and third are the same and are on the same molecule. Okay, because it's normal beta oxidation. This oxidase will also produce trans alpha beta in CoA, so it will do the exact same first step of normal beta oxidation. But the difference is that the electrons from FADH2 won't go. As we said, this may be a trick part, because we said it will go to ubiquinol by ETF. But here we are in the peroxisomes, so we won't be able to do ETC. So we won't be able to. There's no ubiquinol here. So what are we going to do with this FADH2? This FADH2 will transfer its electrons to oxygen. It will transfer its electrons to oxygen, which will cause the H2O2 formation. Of course, the accumulation of such molecules is very dangerous. That's why catalase enzyme will convert it to normal water after. So these are the only differences. The first reaction uses completely different enzyme, which causes the electrons to go directly to oxygen, because it's aslan in peroxisomes, so we can't transfer it to ubiquinol and the plan or the process we know. So it will go to oxygen and then forming H2O2, which then forms water, normal water, by catalase enzyme. And the second and third enzymes are the same and are on the same molecule. Also the thiolase is the same. So we are going to keep using the peroxisomes until what? Until we have fatty acids that are not very long. Then it will transfer to mitochondria and continue its job. So peroxisome is just a temporary stop for fatty acids until they become not very long anymore. So the mitochondria can work on them. Okay, now we have ketone bodies. As you know, degrading the fatty acids produces acetyl-CoA, and sometimes we can produce excess acetyl-CoA. Like for example, the body needed energy that 8 acetyl-CoA is enough to provide this energy. But the body is not like it's not like perfectly it didn't like perfectly calculate how many acetyl-CoA, or maybe it's not about calculation, but maybe there is a delay in the inhibition process, so it produced 10 acetyl-CoA. So let's say the body needed only 8 to produce its energy that's needed, and the body produced 10, for example. So now we have excess of 2 acetyl-CoA. Of course, accumulation of any type of molecule, even if it's very useful to the body, is not good. Any accumulation is not good. So in order to prevent this accumulation, especially that acetyl-CoA have regulatory effect, because acetyl-CoA can signal that there is high energy, so it can have regulatory effect for some reactions. So it is very dangerous to accumulate. So what are we going to do with this excess acetyl-CoA? We are going to convert it to something called ketone bodies. Why do we need to do this step? For 2 reasons. First of all, I just said acetyl-CoA have a regulatory effect, so we don't want this regulatory effect. So ketone bodies doesn't have the regulatory effect. It prevents accumulation of acetyl-CoA, which has a regulatory effect. Ketone bodies doesn't have this effect. Second, acetyl-CoA cannot leave the cell. Like for example, if I need to transfer it to another body part, I can't. It's trapped in the cell. So also ketone bodies can leave the cell. and be transported through blood to reach the target tissue. So actually, we use the ketone bodies to transfer or transport these acetyl-CoA, and then when they reach the target tissue, they will return back to acetyl-CoA. So now, which reactions do we need to know? How to produce ketone bodies from acetyl-CoA and how to produce acetyl-CoA from ketone bodies. Let's go. So as I told you before you approach any pathway or any connected reactions, you need to know what do I want from this? What is my هدف? يعني what do I want? What is the result I am waiting for? So here we have acetyl-CoA. Our result is to produce ketone bodies. What are the ketone bodies? We have three: acetoacetate, beta hydroxybutyrate, and acetone. Okay, so we need to produce these three from acetyl-CoA. Here we are going to use 2 acetyl-CoA. As you can see, 1 second. As you can see, here is number 2. It means 2 from this molecule, and this molecule is acetyl-CoA, which is 2 carbon with CoA. We need 2 acetyl-CoA to start this process. We will merge them. We will merge them with acetoacetyl-CoA thiolase. This is a reversible reaction, as you can see. So in this part, if you know the structure, it will make your life much easier. Like don't memorize it like atom by atom or molecule by molecule, I meant, but just memorize the general structure. So what is acetoacetyl-CoA? Acetoacetyl-CoA is 4 carbons. Here is 2, 1, 2, 4 carbons with CoA. So here we had 2 carbons with CoA, merged those 2 carbons with CoA, so 4 carbons with 1 CoA. Of course, as you can see, 1 CoA is like produced as well. So acetoacetyl-CoA is 4 carbons with CoA. We are going to add 1 more acetyl-CoA. So we need 3 acetyl-CoA for this ketone body formation. By HMG-CoA synthase. From the name, it synthesizes HMG-CoA. HMG came from beta hydroxy beta methyl gluteryl-CoA. So HMG, okay, CoA. And also we are going to produce a second CoA here. What is this HMG-CoA? It is simply 6 carbons with 1 CoA. So we are just adding acetyl-CoA after another until we have 6 carbons with 1 CoA, of course, which means it's still activated, we can use it. Now we are going to use lyase enzyme to cut it, HMG-CoA lyase, which will form 1 acetyl-CoA, this is acetyl-CoA, and acetoacetate. So you are going to ask, we added 1 acetyl-CoA to remove it in the next step? Like, that's literally what happened. We added 1, we removed 1, as you can see, that's the exact same thing. But I'm going to tell you yes, because before we added it, we had acetoacetyl-CoA, which means 4 carbons with CoA. When we removed it, we had only 1 CoA in these 6 carbons. So when we removed it with 2 carbons, the remaining part is 4 carbons without CoA, which is acetoacetate. And if you remember, acetoacetate is one of the ketone bodies. So we had a successful process and we reached ketone bodies. Acetoacetate is simply 4 carbons, and we don't have the CoA. So acetoacetate is actually acetoacetyl-CoA, but without the CoA. So in order to remove it, we added 2 carbons, and then we removed the same 2 carbons with the CoA. Okay? Now we have acetoacetate. It can be converted directly to the other 2 ketone bodies, which is acetone and beta hydroxybutyrate. This reaction doesn't need an enzyme. It is a continuous reaction, and it is irreversible, losing one of the 4 carbons, which that's why we are producing CO2, and acetone is produced. For the beta hydroxybutyrate, we are going to use dehydrogenase enzyme. Of course, beta hydroxybutyrate dehydrogenase. You should have expected that because of the NAD plus, or because it's a reduction, oxidation reduction reaction, and actually this one is reversible as well. And one very important detail also, you have to concentrate on it, which is to memorize the direction. Like here, we use both NAD plus and NADH. Why? Because it's a reversible reaction. So you need to make sure that you understand, that you memorize that acetoacetate 2-bit hydroxybutyrate is the one using the NADH. It is, and of course, when we use NADH, it means we are adding hydrogen. We are need the electron from the hydrogen. We need electrons, so it is reduction this way. Okay, so you need to memorize it and make sure to know the direction, not only the memorization. Because I remember me remembering that we use NADH and NAD plus, but I didn't remember the direction, like which one used the NAD plus and which one the NADH. Okay, so now before we continue this one, okay, so now we successfully produced the 3 ketone bodies. But as I told you, maybe I want to just transport the acetyl-CoA to another tissue. So now these ketone bodies reached another tissue through blood. I want to return it back to acetyl-CoA. What am I going to do? We are simply going to move backward, but there are some differences. First of all, acetone cannot be reversed. So we don't have any reversible reaction, or not reversible, we don't have any other reaction that produce acetoacetate from it. So while once it is acetone, we cannot come back forever to use it as an acetyl-CoA. Okay, this is the first one. Second one, beta hydroxybutyrate. We already know this is a reversible reaction, so we can easily turn back to acetoacetate. The only problem now, if we have acetoacetate, because as you can see, this reaction and this 1 second, it became messy. Okay, 1 second. Okay, so as you can see, if we have acetoacetate, the problem is both of these reactions are irreversible. Like, we cannot go backward simply by the same enzyme and by the same everything. No, we need to change some stuff. And of course, this is for regulatory purposes. You know, any irreversible reaction is probably regulated, or it is due to regulatory purpose. Okay, so ketone bodies to acetyl-CoA. As we said, if we have beta hydroxybutyrate, we will simply go back from the same reaction to acetoacetate by beta hydroxybutyrate dehydrogenase. Okay, so we went back to acetoacetate. Acetoacetate is then going to be transformed to acetoacetyl-CoA. Do you remember the acetoacetyl-CoA? It is this one, acetoacetyl-CoA. I'm just trying to compare them to be easier for you. But because this reaction, which directly converts acetoacetyl-CoA to acetoacetate to acetoacetyl-CoA, is not the same one as the one above, so it has some differences. First of all, it uses beta ketoacyl-CoA transferase. This is the name of the enzyme. Of course, you have to memorize it. It also uses succinyl-CoA as a co-aid or coenzyme A donator. So we need CoA to turn back to acetyl-CoA. So the one which is going to donate for us the CoA is the succinyl-CoA. As you can see, it entered as succinyl-CoA. It left at succinate. It lost its CoA because it gave it to the acetoacetate. So acetoacetate gained a CoA donated from succinyl-CoA by beta ketoacyl-CoA transferase. Now we have acetoacetyl-CoA, which is also going to be converted to acetyl-CoA by the same reaction we had. Like, this is acetyl-CoA. Do you remember acetoacetyl-CoA thiolase? So actually, we can say that beta hydroxybutyrate and acetoacetate, the two that are able to go back to acetyl-CoA, are all of the reactions reversed, except these two are merged into one, going like this, which uses the beta ketoacyl-CoA transferase to produce acetoacetyl-CoA from acetoacetate. I hope that I didn't confuse you with the names because they are very similar. But if you want, just go over it again. It is easy if you understand it, and like it's in a logical sequence, of course. So now we have acetoacetyl-CoA with the thiolase. We have acetyl-CoA. And of course, we have 2 acetyl-CoA. Be aware, this thiolase, we have here 4 carbons. Be aware, this thiolase will cut here, so we have 2 carbons with CoA, and these 2 carbons, the thiolase will add another CoA to it in order for us to have 2 acetyl-CoA, like in beta oxidation, if you remember. Okay, now let's continue with the other types of fatty acid. We have the unsaturated fatty acid oxidation. What is meant by unsaturated? Simply, we have double bond between the carbons other than the CO, the C double bond O. Here, we have in the hydrocarbon chain a double bond. It's called unsaturated. So, is that a problem? Or are we going to proceed with beta oxidation, which is the way we know to degrade them? We have two situations. First situation, if this double bond is between the beta carbon. So okay, so after 3 cycles of normal beta oxidation, for example in oleoyl CoA, we will have this molecule. We don't have to know the exact name for this one. What is the problem here? The problem is, this is the beta. As you can see, this is the beta carbon, and we have the double bond with the beta carbon. This is a problem. So if the double bond is with the beta carbon, this is a problem. We can't proceed with beta oxidation. But if it is with the alpha carbon, it's between the alpha and the beta. So it's with the alpha, it's not between the beta and the gamma. We don't have a problem, and we will proceed. Okay. So there is 2 probabilities. One, that it is between the beta and the gamma, so this is a problem. One, that it is naturally between the alpha and the beta. Then there is another problem, which is that normally in nature, the fatty acids, the unsaturated fatty acids, is found in the cis formation, while the body can only like hydrolyze or destroy the trans formation. So even if the double bond is naturally between the alpha and the beta, which is not a problem for beta oxidation, it will anyway be in the cis formation, and we still can't use normal beta oxidation. So with unsaturated fatty acid, we will add one reaction only, one extra reaction in the normal beta oxidation, in order to change the problem and make beta oxidation viable again. We will use NOL CoA isomerase. And what does the isomerase does? It just changes the formation from cis to trans, and if the double bond is between the beta and gamma, it will transfer it between the beta and alpha. Okay, as you can see here. So instead of cis, we have trans, and instead of double bond with the beta, or between the beta and gamma, it is double bond between beta and alpha, which is no problem for beta oxidation. And then we will continue and proceed with normal beta oxidation, which is NOL CoA hydrase, which is the second process step, and then a resumption of normal beta oxidation. Okay, another type of let's say abnormal, not abnormal, but another type of fatty acid is odd chain fatty acid. If you concentrated, you see that I'm always saying 16 carbon, 18 carbon, 24 carbon fatty acid. But what about if it was 17 carbon fatty acid? What is going to happen? Is there something like this? Yes, there is. So the problem is not with the first beta oxidation rounds, because no problem, we will remove 2, we will have 15, we will remove 2, no problem. The problem is when we get to 5 carbons. We will remove 2, no problem, which is acetyl CoA. We will get 3 remaining. So if you have only 3 remaining, how are you going to remove 2 from it to get acetyl CoA? So if you have only 3 remaining, this molecule is called propionyl CoA. As you can see, 1 carbon, 2 carbon, 3 carbon with CoA. So propionyl CoA is 3 carbon with CoA, which is the remaining of the 17 carbon if we continued beta oxidation for a 17 carbon fatty acid, for example. Here, okay, here we have a very important information. It's mentioned in the book, so unfortunately you will have to memorize it. Okay, But before we get to this information, let's proceed with the reactions first. So propynyl CoA, we are going to need 1 ATP, 1 CO2, and 1 water. Let's break it down. The CO2 is to add 1 carbon to the 3 carbon. We said propynyl CoA is 3 carbon. But what if we want to get 2 acetyl CoA out of it? We need at least 4 carbon, right? Because 4 minus 2 equals 2. So we got 2 acetyl CoA. So we are going to add this carbon. And this addition needs energy from the ATP, and we have water as well. So propynyl CoA carboxylase. From the name, carboxylase is the addition of carbon. This reaction, it is reversible, but it's more derived through this direction. That's why this arrow is shorter. Okay. Here we have dimethyl malonyl CoA. Dimethyl malonyl CoA. The dimethyl malonyl CoA is 4 carbons, because now we added the 4th carbon with CoA. What is the problem here? That the D formation as well can't be seen by the body as well. We need it to be L in order to proceed. So we are going to use methyl malonyl CoA racemase. Racemase changes the D to L. Now we have L-methyl malonyl CoA. And then we will use mutase to change it to succinyl CoA. You are going to say we didn't get. Now we are done. You are going to say we didn't get acetyl CoA. So what? Why? Like we did this? We didn't get acetyl CoA as we normally do from fatty acid. So why do we need succinyl CoA, or why are we satisfied with succinyl CoA? Because we can actually use it to generate energy as well, right? We can integrate it and use it to produce energy. That's why we are satisfied with it, and we don't need acetyl CoA. Okay, now we have the branched chain fatty acid, which is the last special type or different type of fatty acid. Phthalic acid is the most common one. It's found in green vegetables. The most common branched chain fatty acid. Also, here is the same idea. If the branch is attached to the alpha carbon, there is no issue. The problem is when the branch is attached to the beta carbon. So if any special adjustment happened to the beta carbon, it's a problem. Why? Because it's called beta oxidation. That's why we need it to be perfect. So here we are going to if you remember, I told you in the previous odd chain fatty acid, there is an important note I want you to know because it's mentioned in the book. It's actually here in the alpha oxidation. So first, let's start with phentenyl CoA, which is the phthalic acid when it's added to CoA, because we need to activate it as any normal fatty acid. After we activate it with it becomes phentenyl CoA. Okay. The problem here, we have branches, as you can see. These are the branches. If it's in the alpha carbon, no problem, but here it's in the beta carbon. So what are we going to do? We are going to use phentenyl CoA hydroxylase by adding oxygen. Now we have a hydroxyl group, as you can see. So it becomes hydroxy phentenyl CoA, alpha hydroxy phentenyl CoA. Then we're going to use a lyase to remove a carbon. After we removed 1 carbon, we will have prestanal, which is an aldehyde. This prestanal is going to be here, if you can see, it is NAD between brackets P, because here we can use both NAD plus or NADP plus. Okay, so here, as you can see, it is an oxidation reaction because we are losing electrons with aldehyde dehydrogenase. Why aldehyde dehydrogenase? Because prestanal is an aldehyde. So aldehyde dehydrogenase is going to oxidize prestanal to form prestanic acid. Prestanic acid can be proceeded with beta oxidation, because as you can see now the branch is in the alpha carbon. Of course, we agreed that this one is not the alpha because it's the one with the double bond. This is alpha. Now we have no problem with beta oxidation to proceed. The very important note that you have to memorize is in alpha oxidation, which is this one, the first reaction, which is converting the fentanyl CoA to hydroxyfentanyl CoA, is a reaction that requires O2, which is oxygen, alpha ketobutyrate, and the enzyme is catalyzed also by Fe2 plus, which is iron, and you have to remember that it's 2 plus, okay, and ascorbate requiring enzyme. So the enzyme requires ascorbate as well, and it yields alpha hydroxyfentanyl CoA, which is the product, with succinate and CO2. So be aware that the succinate, for example, it is not mentioned here in the picture, but it's written, and you have to unfortunately memorize it as it is. Like this part, just memorization. When it comes to the alpha ketobutyrate and the Fe2 plus with the ascorbate requiring enzyme, which leads also hydroxyfentanyl CoA, succinate, and CO2. So be aware of this point. Here it is a summary. If you remember it this way, you are going, inshallah, to remember all of the other steps easily. When you have unsaturated fatty acid isomerase, you just change the cis to trans, and you put it in the correct location, the double bond. When you have odd chain fatty acid, you will be left with propionyl CoA, which is 3 carbon with CoA. You are going to, after some reactions, you're going to produce succinyl CoA. When you have very long fatty acid, you're going to use the peroxisomes first to degrade it into smaller fatty acid, which can be then processed with the mitochondria with normal beta oxidation. If you have branched chain fatty acid, if the branch is in beta carbon, you will use alpha oxidation, which we just talked about. But if it is in alpha carbon, we will use normal beta oxidation. So it is like the opposite. If the branch is in beta carbon, use alpha oxidation. If the branch is in alpha carbon, use beta oxidation. Now it's practice time. Unfortunately, I didn't put as much questions as I hoped for, because the session otherwise would be very long. It's already long enough. So let's solve these practice questions. Of course, I forgot to mention in the beginning that if you have any question, please don't hesitate to reach me out and ask. Okay, so let's continue. So which enzyme, of course you can pause the video and try to solve yourself. Which enzyme catalyzes the first step of beta oxidation in peroxisomes? So this is a direct memorization question. After you read the choices, you should know it's acyl CoA oxidase. So remember, acyl CoA oxidase is the first, is a special first enzyme in the peroxisome beta oxidation. Okay. HMG CoA cleavage produces also another, like as I told you, you have to understand, so the memorization is easier, but we can't avoid memorization, unfortunately, and you have to have all of these reactions, enzymes, in your mind. So understanding them will make it much easier, and hopefully, inshallah, you remember it in the exam as well, because you understand the process, not just blindly memorizing names. Okay, so HMG CoA cleavage produces acetyl CoA and acetoacetate. Okay, and here, which coenzyme is reduced when beta hydroxyacyl oxidase to oxidizes to ketoacyl CoA? So that's what was I talking about. You need to memorize exactly which one is being used in each reaction. Is it NAD? Is it FAD H2 or FAD? Or what exactly? So here it is NAD. This is normal beta oxidation. And now you continue with the fatty acid biosynthesis and the other part of the chapter with my colleague Shazia. Thank you.
So moving on to fatty acid biosynthesis, This is gonna be in a more sequential, story-like sequence of events, so you guys can keep up whether or not you've learned the lesson properly. These are your main concepts that we're gonna discuss today. We have triggers or regulators of fatty acid biosynthesis, enzymes involved in the process itself, and finally, two small topics: lipoprotein metabolism, this is the endogenous one, and membrane lipid metabolism, that is phospholipids. Okay, starting off. So I want you guys to take a second and guess whether you need high or low levels of these to trigger fatty acid synthesis. So carbohydrates and proteins will be high in your diet for fatty acid synthesis to occur. Why? Because they lead to acetyl-CoA in the metabolic pathways, right? Then you have pyruvate, acetyl-CoA, and citrate. Acetyl-CoA is your main substrate for fatty acid biosynthesis, right? So it's your starting point. So pyruvate leads the formation of acetyl-CoA, right? Pyruvate dehydrogenase complex. And citrate, we will see how it forms acetyl-CoA, or if you guys already know it, I'm not sure. And citrate is also a marker for high energy, right? Hence, these molecules are in high amount. One, for the energy. Two, for the uh, molecules as substrates, mainly acetyl-CoA. Fats should be low in the diet to stimulate fatty acid synthesis, right? It makes sense because if you don't have enough fatty acids in your blood, you're gonna start synthesizing them just like you do for glucose and so on. Then finally, we have insulin. If you think about what insulin does, it lowers the glucose level in your blood. So, what does that do inside the cells? It either directs glucose into breakdown to produce energy or push it into fatty acid synthesis or other biosynthetic processes because you don't want any more glucose around, right? So, how it does that, you can take a look at this. So, insulin mainly carries out its activities by the activity of phosphoprotein phosphatase 2A, which is a dephosphorylating enzyme, right? And if you're thinking where you heard of this before, it's phosphoprotein phosphatase 1, which was involved in carbohydrate metabolism. But anyway, so phosphoprotein phosphatase 2A dephosphorylates a bunch of molecules, including the enzyme ACC. On dephosphorylation, ACC is activated because ACC is a polymer, a dimer to be specific, so it needs to be in its dimer form to carry out its function, right? So insulin binds to the cell, triggers phosphoprotein phosphatase 2A to dephosphorylate ACC, thus activated by making it form its dimer and proceed with the function. Then we have citrate, which also activates this process, not only because it's an energy signaling molecule and it's going to be a precursor for acetyl-CoA, but also it binds allosterically to ACC, meaning it's going to bind it and thus promote its activity directly. Okay. Then we have, on the other hand, palmitoyl-CoA, which is the end product of fatty acid synthesis. So it's simple, direct end product inhibition, right? So it's going to make it depolymerize, create the monomer forms instead of the dimer, and inactivate it. Khalas. Then we have AMPK and pKa. Now, AMPK is a signal for low energy in the cell or tissue. So it's obviously going to antagonize this process. It's not going to let it happen, right? Because it needs energy, but this process uses up energy. So it's going to carry out phosphorylation. AMPK is a kinase, so it's going to phosphorylate ACC and thus inactivate it. Similarly, pKa, which is, I'm sure you guys know, but just to brush up, is stimulated by glucagon and epinephrine binding to the cell. So by second messenger process and so on, pKa is formed and phosphorylates a bunch of other molecules, including ACC. So pKa, AMPK are kinases, so they are going to inactivate ACC. And palmitoyl-CoA is going to inhibit and keep ACC in its inactive form as an end product inhibitor, allosteric, right? On the other hand, insulin activates ACC by dephosphorylation. Citrate maintains it allosterically in its dephosphorylated active form. This bit on the right side is for your simplification if you're confused about, phosphorylation, dephosphorylation, which action goes on how, and so on. Anyway, moving on, we have Okay, so how this process begins before we get to acetyl-CoA, a small brush up. So this is a super summarized version for both whoever has and hasn't learned this. Glycolysis forms pyruvate. Pyruvate enters the cell and forms acetyl-CoA with the help of a specific enzyme we'll be talking about later. And acetyl-CoA we need in the cytoplasm because fatty acid synthesis, unlike beta oxidation, beta oxidation is an energy producing process, so it's going to happen in the mitochondria. But synthesis is going to occur outside, in the cytoplasm, I mean, okay? So acetyl-CoA needs to find its way out. And we know acetyl-CoA is not permeable to the mitochondrial membrane. So what is it going to do? It cannot leave directly, so it's going to enter the TCA cycle or simply just combine with oxaloacetate and form citrate. Again, with the presence of another enzyme we'll be talking about. And now citrate can leave the mitochondria, so citrate leaves at high levels, which is why high levels of citrate stimulates fatty acid biosynthesis. Again, it's kind of like a third reason, I guess. Anyway, citrate enters the cytoplasm, and at high levels, it is lysed. It's broken down back to oxaloacetate and acetyl-CoA. So the merging happens inside the mitochondria with one enzyme, and the other enzyme breaks it down back to the same products, acetyl-CoA and OAA, in the cytoplasm, right? So with this, you have your substrate, acetyl-CoA. Now the enzymes, as we spoke about, step one, pyruvate to acetyl-CoA was pyruvate dehydrogenase complex. Then we have citrate synthase, which is a little less relevant, but just for your refreshing your memory, I mean. Then we have citrate lyase, which breaks down this citrate into the two products that we need. I hope that was clear. Notice how 1 and 3 are activated by insulin. So insulin stimulates the process that occurs right after entering the mitochondria, which is formation of acetyl-CoA, and right after leaving the mitochondria, which is citrate lyase. So it activates these two enzymes. Okay. Now, speaking of the enzymes involved, as we spoke about acetyl-CoA carboxylase, the ACC, we'll talk more about it again later down the line, but it basically carboxylates acetyl-CoA. So we know acetyl-CoA is a two-carbon molecule and it adds carbon dioxide basically to the molecule, forming malonyl-CoA. So this enzyme is the most regulated step. So this is the one that decides whether the process continues or not. It's the very first step, so without it, you cannot continue, okay? Then you have fatty acid synthase, which is a multi-enzyme complex. Your textbook says it's of 7 catalytic domains. So there's 7 regions. If you look at this picture, right? As we said, it's a dimer, so there's one monomer in blue and the other in yellow. So if you look at it, I've marked each of the enzymes as 1, 2, 3, 4, 5, 6. There are 6 enzymes. The seventh catalytic domain, per se, is the ACP, which is not an enzyme. It's simply a carrier, but it is taking part in the catalysis or the reaction. So we say 7 catalytic domains, okay? So ACP is the carrier, unlike CoA, which was used in beta oxidation. I'll get to that in a bit. So what fatty acid synthase does is basically the formation all the way from the beginning, acetyl-CoA. It combines it to malonyl-CoA, then it forms a final product, and then that final product comes back and then merges with another malonyl-CoA, and so on and on and on until palmitate or any fatty acid is formed. Okay, we'll discuss the whole process after this. I want you guys to look at this picture now. This is the comparison between CoA and coenzyme A and ACP. There's nothing you need to memorize about this. This is just for your understanding. So you see here the thiol group, which is very important, as we know, um, is found on this common end. Okay, so you see where the blue lines are. This is where the difference begins on the right side between ACP and CoA. Otherwise, everything to the left, including the thiol group, is common. Okay. Then, as I said, it's a dimer, and notice that Ks, a particular enzyme that we'll talk about, has a thiol group that I have added to it. So, considering that it has a thiol group, what do we think is the amino acid involved in this enzyme? It's going to be cysteine. Okay? So moving on to the formation of all the way from acetyl-CoA to palmitate, I have summarized in one single slide. So this is super condensed. Ideally for those who have learned it already, but those who haven't, we'll go into more detail in the next slides, okay? So we start off, firstly, this enzyme is a monomer. Clearly, there's only one unit of each enzyme and not two in the complex, right? All right, so we have acetyl-CoA in the cytoplasm. It's going to do one of two things. Either convert to acetyl ACP or become malonyl-CoA. So what's happening in A, this process, is the acetyl is being transferred from CoA to ACP. Okay, so that occurs, and then acetyl-CoA to malonyl-CoA we discussed happens in the ACC, CoA, acetyl-CoA carboxylase, right? Now, malonyl-CoA, after it's formed, is going to, again, the malonyl is going to be transferred to the ACP. So the name of this enzyme is malonyl acetyl transferase, all right, because it's transferring these two groups from CoA to ACP. So it's transferase. And this enzyme is actually found inside the FAS complex, but I kept it here for the simplicity of differentiating what goes where, right? So in the MAT, acetyl ACP is formed and malonyl ACP is formed. Now, acetyl ACP, as you know, is acetyl connected to ACP by a thioester linkage by a sulfur, right? So what's going to happen is acetyl leaves from the ACP, leaves the ACP along with its thiol group, so ACPSH leaves. Acetyl goes all the way to this enzyme from MAT. So MAT, assume it's like here, MAT, to the ketoacyl synthase enzyme, which also has the thiol group. So it's going to thioesterify with this enzyme instead of with ACP. So ACP is lost. Acetyl forms the thioester with the synthase, forming the complex that we call acetyl synthase. So this is not a separate enzyme. This is still KS, esterified, thioesterified with the acetyl group, right? Why is this acyl? Because we'll see about that later, okay? So acetyl synthase. Now that we have acetyl attached to the synthase enzyme, malonyl ACP is going to come from MAT again, the transferase enzyme. As soon as it's formed, it's going to merge with the acetyl in the acetyl synthase. So acetyl, two carbons, thioesterified with synthase. Malonyl, three carbons, thioesterified with ACP. What's going to happen? Malonyl is going to lose a carbon dioxide minus CO2, and it's going to merge. It's going to keep its ACP, by the way. It's going to merge with the acetyl group over here and make the acetyl cut the bond with the synthase. So you have the ACP from the malonyl and the acetyl group after decarboxylation and the acetyl group from the acetyl synthase. خلاص, okay? So what forms is an acetoacetyl ACP. Hence the name of this enzyme being ketoacyl synthase. We'll get to this in more detail later. Anyway, so you have acetoacetyl ACP formed. It's going to move on to the next enzyme. As we see, it's a ketoacyl ACP. So this is a acetoacetyl, right? Acetoacetyl ACP enters the ketoacyl ACP reductase to be reduced. It's going to get reduced and then form beta hydroxy. So the keto group is converted to hydroxy on reduction. So hydroxyacyl ACP enters this enzyme and um, comes out dehydrated to form an enoyl ACP. Now, why is that? Hydroxy group is being dehydrated, right? So you're forming a double bond between the carbons now. So the carbon double bond is the ene part of enoyl, and the oil part is basically the acyl, the thioesterification to the ACP. Okay, I hope that was clear. Um, Hydroxyacyl enters this enzyme, comes out as enoyl. Enoyl enters this enzyme and gets reduced again. Why is the double bond being reduced? So you can have a straight, simple, saturated hydrocarbon chain, right? So what do you think is the name of this product? We have four carbons in a straight chain, thioesterified with ACP. So that's butyryl ACP, four carbons, okay? So what's going to happen here? Butyryl ACP forms what we call acyl ACP. Acyl is just the template name, right? Butyryl is a form of acyl, just like acetyl. So that's why I've named all of these acyl, right? So now what was acetyl ACP has gone through all of this and become butyryl ACP. Now butyryl ACP enters again, becomes butyryl synthase. Malonyl ACP, another block again, joins this and forms a 6-carbon product, which then gets reduced, dehydrated, reduced, and so on and on and on. 6 elongation cycles after the first set of reactions. So a total of 7 cycles of the same process, ending up in the production of palmitoyl ACP. Now, palmitoyl ACP, as we spoke, palmitate is the final product, so it's going to enter the thioesterase or thiolase, same enzyme, different names. Thioesterase cuts the bond between the acyl and the ACP. This is where the S is actually removed and given over to the ACP, right? ACP receives the thiol group after being worked on by the thiolase. This acyl here is technically our palmitate, since that's what we're talking about, so it leads to the product formation. And then we have ACP with its thiol attacking malonyl-CoA in the MAT again, so the cycle continues. I hope that was clear, and this is the net products and reactants involved in the process. Very important. You do need to know the numbers. We'll go over it later. 8 acetyl-CoA because you're starting with acetyl-CoA in ACP and in the formation of malonyl. So you have starting product as acetyl ACP, and every next product formed is combined with another 2 carbons from malonyl-CoA. Now, more in depth on the process. So as we said, acetyl-CoA is found in the cytoplasm, is converted to acetyl ACP by the malonyl acetyl transferase. It transfers the group of malonyl or acetyl from CoA to ACP. The moment it has ACP, it's ready to be worked on in the enzyme that is Ks over here. Okay. Now acetyl-CoA gets converted to malonyl-CoA with the help of acetyl-CoA carboxylase. So it uses carbon dioxide, but the problem is, you see carbon dioxide everywhere. It's a very stable compound. So it's not easy to just casually incorporate it into acetyl-CoA. So what do you need? Energy to activate the carbon dioxide and make it active enough to attack or be attacked by the malonyl-CoA and form, sorry, by acetyl-CoA and form malonyl-CoA. So remember, acetyl-CoA carboxylase has biotin as its coenzyme slash cofactor. So acetyl-CoA itself consists of 3 different catalytic regions. One is your biotin carboxylase, where biotin is carboxylated, then carried by biotin carboxyl carrier protein in its carboxylated form, right? Biotin is carboxylated and then it's carried by the carrier protein to the carboxyl transferase site where it decarboxylates and transfers the carboxyl onto the acetyl-CoA to form malonyl-CoA, right? So carboxyl transferase CT is your site of the substrate. BC is where it gets carboxylated, the biotin gets carboxylated, and BCCP is the arm, okay? Now, ATP is required to activate the carbon dioxide, and how this happens, this is mostly for your information. I highly doubt you'll be tested on it, but this is your carboxyl group attached to the biotin in green, and this is your substrate acetyl-CoA, and it has been deprotonated by the enzyme itself. So this is carbanion, and this is a carboxyl group with a partially positive charged carbon. So this carbon is going to attack this one and thus form malonyl-CoA. So more in depth about acetyl-CoA carboxylase, we know it uses ATP, carbon dioxide, 3 regions in the enzyme, and a biotin coenzyme to form malonyl-CoA from acetyl-CoA. It's the rate-limiting step. Yes. Now, the very important part here is ACC1 and ACC2. So, ACC1 has one main function, that is biosynthesis of fatty acid, thus found in lipogenic tissues. ACC2 also does the same function but is found in oxidative tissues, so it has a second function, that is regulation of oxidation of fatty acids in the mitochondria, so it's found on the outer mitochondrial membrane. Liver, as always, the metabolic boss of sorts, has both enzymes. Okay, so moving on to what happens after acetyl ACP and malonyl ACP are formed. We already spoke about ACC 1 and 2. Acetyl ACP binds with, as you can see here, the ketoacyl synthase enzyme with the thiol group and forms acetyl with the synthase enzyme. Okay, it's esterified with the synthase. And ACP is released with its thiol group. Now, acetyl synthase is formed, malonyl ACP is formed, so this is within the ketoacyl synthase. This is the synthase, this is the acetyl. So what happens? Malonyl ACP is going to get decarboxylated, right? And the rest of it, the acetyl ACP, basically, right, is going to merge with the acetyl group of the acetyl synthase, meaning the thioester group with the synthase is cut off. So you have aceto, acetyl, ACP, and that is your product. Okay? So now why this enzyme is called beta ketoacyl synthase is because if you look at the final product, right? Firstly, beta ketoacyl is the common name. It's like a template name. So at the beta position, there is a carbonyl or a keto group, and it's an acyl ACP. So it synthesizes this molecule, and hence the name. Okay, and it catalyzes the condensation reaction of these two molecules, right? This is the variable acyl synthase. In this first stage, it's the acetyl synthase, and malonyl always continuously gets added and is constant. Okay, so this is aceto, acetyl, ACP. All right, so we just remember this is how the condensation happens. The thiol of the component attached to the synthase is removed and it's bound to the malonyl that is decarboxylated. So it's an addition of an acetyl group, technically, even though it's the malonyl ACP that's getting added to the acyl group that is forming. Okay, so after this stage, now that we've formed, condensed our acetoacetyl ACP, which is this molecule exactly, now our main goal of the next few steps, the next three steps, is to convert this carbonyl group into a CH2. Why? Because your final product is a palmitate, which is a completely saturated fatty acid. So you don't want any double bonds, no nothing. Just CH3, CH2, 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 and so on, right? So we're going to look at that now. This is one step of one cycle of elongation. So it consists of a condensation, a reduction, a dehydration, and another reduction. So you want to convert, as we said, carbonyl to CH2. So this guy here, the beta ketoacyl ACP reductase, is going to use an ADPH to reduce. So it's going to add the hydrogen here. This is what I spoke about. So at the beta position, the carbonyl is being converted to an alcohol group. So it's going to be beta hydroxybutyryl ACP. Hydroxy at the beta position, and it's butyryl because there's now four carbons, right? Initially, they called it acetoacetyl because there's two carbonyl. Now it's a single is being observed as a whole. So that's why the name becomes butyryl, hydroxybutyryl, okay? Now that you have OH, what do you want to do to get rid of the OH? Dehydrate. Once you dehydrate, you end up with a double bond. Again, you don't want it. So what do? Reduce. Reduce using the ene oil ACP reductase. The ene is the double bond, oil is the thioester, or any esterification makes the compound name end with a YL, okay? So that leads to the formation of your saturated acyl ACP. So you have butyryl ACP in this case. Okay, so butyryl is gonna go back as in place of acetyl ACP. So that's why I keep saying acyl ACP, because now in the second stage of elongation or second stage of the process, second cycle, butyryl is gonna come back and replace this. So it's gonna be a butyryl synthase and form citral ACP. It's not mentioned in your book. Don't worry about that detail. So that concludes the elongation part of the synthesis. It's a condensation, reduction, dehydration, and reduction again. All right, and finally, now that you have butyryl ACP and then it's being cycled back and back and back, added with malonyl CoA another 6 times after butyryl ACP is formed, right? So that's a total of 7 times going through this entire thing until palmitoyl ACP is formed. Once palmitoyl ACP is formed, it's going to enter the thioesterase in the enzyme complex or thiolase and break off from the ACP and form your final palmitate or fatty acid. Okay, now keep in mind that this is elongation that occurs as part of your synthesis of the fatty acid, right? Completely as part of the process. There's also elongation that occurs to the palmitate that's formed and done. So you have fatty acids circulating around in your body as either dietary fatty acids or produced fatty acids, anything. Those can enter back into the cell depending on demand and be elongated or desaturated, as we'll see later. So these processes occur in the ER. So that is separate from the synthesis that occurs in the cytoplasm. I hope that's clear. We'll look at it again anyway. So coming back to this diagram, we have acetyl-CoA formed in the cytoplasm, and like I said, fatty acid synthesis forms palmitate. Palmitate can either go to the mitochondria or the ER for further elongation or modification, but most of the time it's ER. All right. So this is what we spoke about, elongation after synthesis, right? So we said cytoplasm and ER. In biosynthesis, it's ACP that's being the carrier molecule, right? In modification, that is after the palmitate is formed and it's taken to the ER to elongate again, it's esterified to CoA again. It's not ACP, right? Then 7 cycles of elongation altogether in net for palmitate synthesis, but in modification, it really depends on the demand, right? So it's just to put that into perspective. And it involves 4 enzymes, as we said, synthase, reductase, dehydratase, reductase, and then finally, in the last step, once you're done with everything and you just want to release the ACP, you take the thiolase. Now we have desaturation, which is also very much closely linked to elongation in the ER. So this isn't really that important. You just need to know that it uses NADH, unlike the biosynthetic process that uses NADPH, right? So it uses NADH electrons from it. The electrons transferred from NADH to cytochrome B5 reductase, an enzyme that reduces, thus gives electrons to cytochrome B5. B5 gives them to desaturase, another enzyme, which is going to give those electrons to an oxygen molecule or oxygen molecules and activate them because now they're going to be even more electronegative and they need more positive charge. So they'll take away the proton. Now, why do you need to take away the proton? Because think about it. How do you desaturate a molecule? Say, for example, you have a CH3CH3 and ethane, right? And you want to create a double bond between them. So you want to add another bond. How would you do that? You would remove 1 proton from each side, leaving 2 electrons with the carbon alone. The electrons come together and form a second bond, which is the double bond, right? So moving on to regulation of fatty acid synthesis, we have AMPK and hormones being mainly short-term regulators. AMPK, as we said we talk about, is AMP activated protein kinase. So one thing to take from that is it's a kinase, so it carries out its functions by phosphorylation. Secondly, it's AMP activated. So in AMP activation, there's 2 ways in which it does it. One is allosterically, it binds AMPK and stimulates its function. Secondly, the high levels of AMP as compared to ATP signal low energy in the cell, signal kinases that phosphorylate one another and another and another until they finally phosphorylate AMPK and activate it. Okay, I don't think this is that important, but just for your information. High AMP can be caused due to stressors like malnutrition, hypoxia, heat shock, and so on. And the more important concept of AMPK is that it needs more energy, right? So it's going to promote catabolism. How? By stimulating phosphorylation. So phosphorylation would stimulate processes that produce energy and inhibit processes or enzymes that are involved in using energy, like ACC, glycerol 3-phosphate, acyltransferase, and so on. Examples, I doubt they're important, but it's your call. Beta oxidation stimulated by AMPK's activity on these two enzymes. Hormones, on the other hand, we have insulin, epinephrine, and glucagon. So insulin, as we said, it dephosphorylates and activates all of the above, all of these, PP2A. PDHC and citrate lyase are the ones that we said work as soon as the molecule enters and leaves the mitochondria. GLUT4, it stimulates increased glucose uptake, so it can be converted again to pyruvate, to acetyl-CoA, to fatty acid synthesis. Now, HSL promotes lipolysis. So what I want you to understand here is insulin dephosphorylates to reduce blood sugar content. HSL promotes lipolysis, and lipolysis to a certain extent is kind of a gluconeogenic process, so insulin and HSL have opposing activities. So when insulin dephosphorylates, it's going to depress the activity of HSL, right? There's that. And on the other hand, you have epinephrine, which does the exact opposite. It phosphorylates and thus activates lipolysis carried out by HSL for two reasons, the need for glucose and or the need for energy. Glucagon, on the other hand, is involved more in activating fatty acid oxidation. Moving on to long-term regulation of fatty acid synthesis is by gene expression. So to make this very simple and avoid any complication, so we have two terms, response elements and transcription factors. What I want you to know is response elements are basically what we know more familiarly as promoters, right? They are a DNA sequence region on a specific gene or before a specific gene that codes for a specific protein or enzyme, right? So the point of this is, okay, let's look at this, right? You have a promoter region in this particular section of DNA. So here is where the proteins called transcription factors will bind. What do? They summon more molecules that are involved and required for transcription to start. Now, why is this important? Because we have two examples of transcription factor classes called SREBPs and PPARs. May or may not need to know the name, I am not sure. Basically, they are these classes of proteins that are involved in regulating the transcription of genes that end up in the production of enzymes or proteins that are involved in fatty acid synthesis or metabolism altogether. So, for example, here, again, you don't need to know this one, I highly doubt it. High levels of glucose would activate a specific transcription factor called carbohydrate regulatory element binding protein. It's a transcription factor, so it binds to the promoter region of the gene that leads to the production of enzymes like ACC and the fatty acid synthase complex. Okay, so I'll give you guys a second to figure out whether these are true or false. Okay, so the first three are pretty self-explanatory and very theoretical, so I'll leave it at that. Starting from the fourth one, starting from malonyl-CoA to palmitoyl-CoA, from 6 ATPs are used. Okay, so trace back to where you need ATP. ATP is involved in only one step, which is your rate limiting step, which is acetyl-CoA to malonyl-CoA. So that happens once in the start, the first stage that we looked at in the entire diagram, and then this next 6 processes of elongation also require 1 ATP each. So it's one in the first stage, and then every next stage of elongation, which is the 6 elongation cycles. NADH inhibits beta hydroxyacyl-CoA dehydrogenase. Yes, NADH is a signal to a certain extent for high energy, and if it inhibits, it's going to inhibit the enzyme involved in the process that makes more energy, right? Makes sense. And not to confuse yourself with beta hydroxyacyl-CoA dehydrogenase versus ACP dehydratase. Okay, so you need to be careful when reading those questions. Acetyl-CoA is used in fatty acid synthesis. Yes, it's the starting point, not an intermediate. Intermediate will always be ACP products. This is very important. Then you have a net production of carbon dioxide in fatty acid synthesis. No, because as we see here, ACC carboxylates, while in KS, the synthase, where the condensation happens, the CO2 is released from malonyl, leading to the formation of acetoacetyl ACP. Okay? Then finally, we have Palmitoyl-CoA promotes ACC depolymerization. Yes, it's an end product inhibitor. I hope that was clear. So given the past questions, I'm sure you guys realize that you do need to know all of these numbers very well, and hence why we explain in so much detail so you understand what's going on and you don't need to memorize and get stuck no matter how the question is phrased in the exams, okay? So 8-acetyl-CoA, why? Because you have, all right, say you have bricks, okay? Your bricks are acetyl-CoA. So your first stage that we see the whole diagram of in the textbook is of putting acetyl-CoA, which converts into acetyl ACP, and malonyl-CoA, which also comes from acetyl-CoA and then is converted to malonyl ACP. You're putting those two bricks together. That is your first cycle. After that, you have the butyryl ACP, your product, coming back and being added to another molecule of, or another brick of malonyl ACP. So this is where elongation begins. So this is the first stage of elongation after you have the two bricks put together in the form of butyryl ACP, and then you're adding another brick every next 6 elongation cycles. So plus 6 malonyl ACP, and the initial two acetyl-CoAs that are converted to malonyl ACP and acetyl ACP. I hope that was clear. So that explains why acetyl-CoA is 8. NADPH and ATP are, okay, everything after condensation happens 7 times. Everything before condensation you need to think about, okay? So after condensation you have two reactions of NADPH. So you have 7 times that it happens, 1 in the first stage and then 6 in the next 6 cycles, and that's a total of 7, and there's 2 reactions of redox, so you have 14 NADPH used. Then ATP, because it's used in only one reaction and it's the malonyl CoA formation side, so that also happens 7 times. Last but not least, you have 6H2O. Where is the one other H2O that we need to consider at 7? It's used up in the last stage of synthesis, that is thioesterase, right? So H2O is used in cleaving the thioester bond, right? And this again is a very concise table. It is needed, it is going to be tested on, but it's everything that you learned so far, so go through it if need be. Okay. Second to last, we have lipoprotein metabolism, but this is the endogenous version. So this slide is pretty self-explanatory, so I'm going to leave it to you guys. The most important part, however, is the apolipoproteins, so I'm just going to go over that. ApoB100 is found in the nascent VLDL that is endogenously produced on the liver cells, in the liver cells, and it enters the blood, receives ApoE and C2 from HDL, and becomes mature VLDL. And C2 that it receives is used to be recognized by LPL near the target cells, not in the target cells. Then it goes into IDL. So I'm using these colors to denote where these are found. So VLDL is in the blood, IDL is in the liver, LDL is in the target cells, okay? And IDL uses ApoE to be identified or recognized by hepatocytes and those broken down. And LDL uses ApoB100. So the last stage, LDL uses the apolipoprotein that came at the very beginning, like it was produced with it. Okay, that's it. Now we have phospholipid metabolism, which is a form of membrane lipid metabolism. And we have 3 specific types of phospholipids that we're going to talk about the synthesis of today. First off, we have ethanolamine. Okay, I'm going to consider this your parent group. We'll talk about that later. So when you look at it, you have ethanol, OH, CH2, CH2, and then amine. So it's very direct, okay? So this is the structure. Now you want to convert ethanolamine as it enters the cell into this structure, which is phosphatidyl ethanolamine. Now, what is phosphatidyl? It's firstly you have the diacylglycerol in the yellow, right, along with the esterified components, along with the phosphate makes it phosphatidic acid. Okay, so it should be phosphatidyl with ethanolamine esterified to it. But in the process, we use diacylglycerol and not directly phosphatidic acid. So where does the phosphate come from? That's why we have the whole elaborate process on the right side. So let's look at it. You have ethanolamine. As soon as it enters the cell, it's phosphorylated by a kinase. So it forms phosphoethanolamine. So it has a phosphate at the end of it. Now, I want you guys to keep the enzymes aside for now. We'll come back to them later, and I'm telling you, trust me, it's very easy, very simple. Don't worry about it, okay? So you have phosphoethanolamine. The moment it's been phosphorylated, your mind should go to the immediate next step, which is addition of CTP to form a CDP ethanolamine. So if you want to go into the mechanics of what's happening, you have cytidine triphosphate, right? Three phosphates and a cytidine. You already have one phosphate on the phosphoethanolamine. So all you do is add the cytidine and the phosphate, one phosphate. This unit is called cytidyl, is being transferred onto the phosphoethanolamine component, right? So that leads to the formation of CDP ethanolamine. What happens to the remaining two phosphates? They leave as pyrophosphate. Okay? Simple as that. Now, CDP is what you need attached to the molecule to have it react and form your final phosphatidyl component. Okay? So without CDP, your molecule is not going anywhere. So CDP ethanolamine combined with diacylglycerol will form phosphatidyl ethanolamine as you see on the left side. So the phosphate comes from phosphoethanolamine, which is then converted to CDP ethanolamine. Okay, so break it down. The name is only based on the big picture. It doesn't tell you where what is coming from. Okay, so that is it for phosphatidyl ethanolamine formation. Next, we have choline. You'll be happy to know choline is exactly the same process as ethanolamine. Okay, so try to brush up with me. Choline enters the cell, gets phosphorylated, gets CTP added to it, and becomes CDP choline and releases pyrophosphate. CDP choline reacts with diacylglycerol to form phosphatidyl choline. Okay, that's what you see on the left side for ethanolamine. Now, don't look at the enzyme name. Okay, it's just phosphoethanolamine reacts with CTP that's added to form CDP ethanolamine. And what's being transferred from CTP? You have a cytidyl transferred to the phosphoethanolamine. So you have CTP, the donor, phosphoethanolamine, the recipient, and the action that is going on, which is the transfer of cytidyl from one to the other. So going back to where the reaction happened, right? So CTP, phosphoethanolamine, cytidine and a phosphate transferred onto the phosphoethanolamine. So CTP, phosphoethanolamine, cytidyl transferase. Okay, you just need to know the names of the components and then you'll figure it out. Okay, next step you have CDP ethanolamine, 1,2-diacylglycerol. What is being transferred? Phosphoethanolamine onto the diacylglycerol. So that's it. CDP ethanolamine, 1,2-diacylglycerol, phosphoethanolamine transferase. Okay, so it's a kinase and two transferases, especially for PE and PC, ethanolamine and choline. Okay, now we have Serene. The name gives me a very queen kind of energy, a very arrogant queen. Now, why am I saying this? Is because this one doesn't want to do anything. It doesn't want to get phosphorylated, doesn't want to do any of the work. It's just there as it is. What happens is it makes diacylglycerol do all the work. So going back to the structure of diacylglycerol, right? Diacylglycerol, when it gets phosphorylated, forms phosphatidic acid, right? Phosphatidic acid, just like what happened with the molecules we had here, is going to happen exactly the same way to diacylglycerol. So first it's phosphorylation, then you have the addition of CTP, so it becomes CDP diacylglycerol. So think about it. You have a phosphate and then another phosphate and then a cytidine. Okay, so when you put the cytidine and the two phosphates together to call it CDP, what's left is a diacylglycerol. So that is how CDP diacylglycerol is formed, and then it directly reacts with serine. Okay, so the enzyme involved would be diacylglycerol kinase because it's getting phosphorylated, and then again, phosphatidic acid cytidyl transferase. Obviously, you can say CTP, phosphatidic acid cytidyl transferase, no big difference. All right. Now we talked about how each of these, PE, PC, PS, are respectively formed from their initial molecules, ethanolamine, choline, and serine, right? Now, there's a way in which each of these can be interconverted with each other. Okay, so you have that's where this structures comes in. Okay, so I want you to look at, as I said, ethanolamine being the parent group, so it has a hydroxyl, two carbons, and an amine. Choline has the same thing, but instead of the amine, it has three methyl groups instead of three hydrogens. That's it. Serine, same as ethanolamine, only difference is it has an acid group, a carboxylate group, attached to the alpha carbon because it's an alpha amino acid. All right? So this is for your understanding because this will help you understand how these are converted to each other. I'm not sure if this is very important, but again, it's biochemistry, you never know. So phosphatidyl ethanolamine to choline would be by, guess the reaction that's involved. It should be methylation. There's three stages of methylation that occur, and each of these processes has an enzyme that I will not go into. So the enzyme doesn't directly use any methyl groups that are just like, hanging around or anything. No. There is a specific molecule in our body called S-adenosyl methionine, a molecule that contains a free methyl group, so it can provide it for methylation reactions. Okay. Next, PE to PS. So you have ethanolamine converted to the same thing with an acid. So you'd think carboxylation. But why bother? Because carboxylation, as we know, it's ATP dependent. It's very energy consuming. So we're not going to do that. What are we going to do? It's like PS is demanding to be made from PE. Like I said, very arrogant, right? So it's going to make what you call a head group exchange happen. So what's the head group? If you look over here, right, you have what is phosphatidylcholine. Okay, assume this is ethanolamine, all right? The polar head group is anything that's associated with the phosphate. So this region is the head. So what's going to happen? Serine is going to be hanging around, as always, lazy, arrogant, and it's going to make PE come around and do a head group exchange. So basically, the ethanolamine leaves and serine comes and replaces and becomes phosphatidylserine. Khalas, that's it. It's the head group exchange. But PS to PE is where we bring about the carboxylation, in fact, decarboxylation reaction. Pretty simple, right? And if you do need to remember where it occurs, it's the mitochondria because it's involved in respiration, gas exchange, and decarboxylation produces a gas, right? So think of it that way. So, lastly, diacylglycerol has three fates. One, it directly combines with a CDP derivative to form PE or PC. Secondly, it goes under fatty acid esterification by diacylglycerol acyl transferase, as we know. And lastly, it undergoes a phosphorylation and addition of CTP by itself to combine and form PS. That is it. Now we have some questions. I'd like you guys to go through it. I'll give you a second. So one of the products of PDHC is the thioester of acetate. So for those who are not sure what the difference is, acetate or acetic acid with the OH, ester when the H is replaced by a substituent, thioester is when there's a substituent attached to an S instead of an oxygen, sulfur instead of oxygen, thiol, right? That's it. Okay, so next question. Which of the following enzyme is not found in the process of fatty acid synthesis? It is beta-hydroxyacyl-CoA dehydratase. Why? Because it says CoA, not ACP. If it was ACP, the answer would be correct. Next, what is not required in fatty acid synthesis? None of the above. We use carboxylation in acetyl-CoA to malonyl-CoA, so that's pretty much all of the above options. Carbon dioxide requires biotin and converts acetyl-CoA to malonyl-CoA. What is true of the enzyme that catalyzes a reaction from acetyl to malonyl-CoA, which is ACC? It uses ATP, not ADP. Produces carbon dioxide? No, uses carbon dioxide. AMPK activates it? That doesn't make sense because it uses ATP, so AMPK should inactivate it, so it's none of the above. What is the role of lipoprotein lipase? I think you guys have already covered this in the first half, but anyway. Endocytosis of VLDL? No. Endocytosis of VLDL doesn't happen. LPL just performs its action outside of the cells and breaks down the triacylglycerols into fatty acids and glycerol. Hydrolyze the triacylglycerols and lipoproteins? Yes. So that's it. Thank you very much. Do not hesitate to reach out if you need any help or if you have any questions regarding this lesson. I hope this was helpful. That's it. Have a great day.